So yeah, I just wanted to go through a little bit more in detail about what you would communicate with Sam whenever you could give him a call. We'll just click through these since we're kind of behind. So what questions do you want to answer? What comparison types do you want to do? Are they tissues? Are they cell culture? Are they pull downs? Any of those types of things. That's the first thing he's going to ask you about what you want to do. And then how many replicates do you need? Like Dennis said earlier, we like to have at least 5 replicates for most things, especially tissue culture, you may be able to get away with 3 replicates. But anything to do with animals or pull downs or things like that have a little more variability in them, we really ask that you have 5 replicates, just because you're going to have more variability. The statistical significance is not going to be there for as many things as you want it to be. But if that's all you have, that's all you have, and we'll still work with that. Are you looking for any of the post-translational modifications? So things like phosphorylation, ubiquitination, acetylation, just any of those that were in that big circle right there, because that is going to determine what methods we use, how much protein you're going to need, and if you need to do those enrichments before you send us the samples. So we just discussed this pretty much with the TMT versus DIA. It's going to depend on your biological question. Most of the things we're going to push you towards DIA at the moment, just because, truthfully, we get better data on the astral than we do with the TMT at the moment. So we're probably going to push you that way, unless, like Dennis said, there's a reason why you need to do TMT. So how much protein do you need whenever you send, or how much tissue or whatever? As of like 5 years ago, you would need these amounts over here on the left. So you'd need 10 to the 6 cells, 100 milligrams of tissue, or 50 microliters of a serum or plasma sample. This is from 2024. If you go to 2026, it's a little bit less than this even. We can get away with 10 to the 4 cells. 10 milligrams of tissue, or down to 10 microliters of serum or plasma. We like to have a little bit extra, because if we need to go back and redo something, if there's something wrong with the sample when we run it, we'll go back and redo it. So we like to have a little bit extra than what we need. Around 2027, we should be able to go down to these levels, just with the methods that we use. And the single cell, we're starting to work on that, but it's a little ways down the road still.
And we're going to use it a front loader then?
Yeah, I guess. I don't know. This is one of Sam's slides. So I'm not really sure where that came from, but yeah, I guess we went from needing, bucket loads down to a smaller amount in just like 6 years. So
can I ask a question? If it's human tissue, so you say tissue sample, if it's a muscle sample.
If it's a muscle sample, yeah.
Is that the weight of the sample?
The weight of the sample, okay. Yeah.
Thank you.
So when we process it, we'll be able to get enough protein out of it. And I will tell you that this is what we need. We like to have a little extra if possible, but we can go down to that. And so, sure, go ahead. No, you're fine.
Do you mind just going back one slide so I can take
we will also, you'll have all these slides available to you.
You haven't sent out the box.
Yeah, you'll get all of these slides for both days.
I can do that right now.
Very soon, actually. So then once you decide what type of sample you're going to have, we'll decide what instrument it needs to be run on. So the smaller amounts of protein we can run on these 2 instruments here. If it has to be phospho-TMT or TMT, they will be run on the Orbitrap Eclipse with the offline peptide fractionation for the labels, or to fractionate it. Another key factor is how much instrument time is required. Like we just said, some of the instruments you have to run them longer than the others, and you'll still get the same amount of information. So we try to keep those costs about the same. But you will never get the same level of depth on the explorers as you would the astral. So in the end, if it's worth your money, then you can run it on the astral, depending on the sample type. So this is just another explanation of that. So down here with the astral at 30 minutes, we can get around 8 to 10,000 proteins identified, where it would take 24 hours on the eclipse to get 8,000. So once you have that consultation with Sam, and you decide on a project that you want to do, he will send out a project summary that looks something like this. And it'll have a price quote down at the bottom. Don't really pay attention to the actual numbers here because this isn't what we're actually doing yet, because this is for phospho-DIA. But basically it'll have the date, project identifier, which would be your last name, and the date and what type of project it is. That way we can go back to it later. If you need data, if you need it reanalyzed, or if you need methods or anything like that, it'll be linked to that identifier. And then it will have the PI's name, the institute, the types of sample that you want to send in, the number of samples, and then what organism it's from. Then it'll also have the project workflow. So this is what we're going to do once we receive your samples. And the sample groups, and then comparisons. I don't have it listed here, but well, there's only 2. So we're only comparing treatment versus no treatment. Then once he sends you that and you're ready to send in your samples, he will send you an empty sample list template where we'll need the sample number, the sample name, sample groups. So these are treated versus untreated. The type of sample. So these were cell pellets, human organism, the cell line, if you have it, the sample buffer, if they are in a buffer, which with the cell pellets, they wouldn't be, but with this one, this is the cell buffer or sample buffer that we use. How much sample there is and the sample concentration if you have it. So if you've already done a BCA, we're still going to do another BCA, but that way you have an idea of what you sent in or what you think you sent in. And then if you have tissue, the sample mass, so that would be where the milligrams of tissue would be. And then any other pertinent notes that you might want to add in there that might be helpful to us. As in, if you isolated these samples on different days or something like that, we need to know that because it can cause batching effects. So once you have that and you send it back to Sam, then he will give you the information to send the FedEx package into us. So once we get your samples, well, this is what we like to have you do before you send your samples. How you prepare your samples actually determines how good of experimental success you have. So the more consistent you are with your samples, the better results we can have in the end. We like to have the samples in HPLC or LCMS grade solvents and reagents. Use 1.5 mil propylene tubes. Don't autoclave them though. It can cause contamination of the samples with the plastics. Also, label the tubes clearly as possible, because whenever our sample prep people get them, they need to line them up with your sample list so they can identify which ones are which. Yeah, make sure that the adhesive labels should be cryo resistant if you put them on top of the lids here, because sometimes we'll get samples in and they've been on dry ice and the labels have fallen off the top. So just make sure that those are cryo resistant. And avoid freeze thaw cycles before you send your samples. So this is just an example of the 1.5 mil tubes and how we get them and if they're labeled clearly. This is really the best if you can label them that way. Just with a single number that corresponds with your sample list. If we get samples in these other types of tubes, then our sample prep people have to move them into the 1.5 mil tubes. So that takes more time, more of a chance of something going wrong. So we just like to have them in the 1.5 mil tubes if possible. So like I said, with the sample list, you have numbers or names on the sample tubes, match the list. The sample numbering system, if you just do 1, 2, 3, 4, that's great. And then you can put in the details of your different types of samples there. We like to have them ordered by group and then by replicate. That way, when we do the bioinformatics, it just makes everything a little bit easier for them also. And then we randomize them before running them on the mass spec, and then we'll randomize them, and then they'll be reassociated with their appropriate sample number after that. So like I said, with the cultured cells, we like 2 to 5 times 10 to the 6 cells. Fresh frozen tissue samples, 10 to 100 milligrams. In the tissue sample, actually, I think I'll talk about this later, but in the tissue samples, we like to try to have them as uniform as possible. And say you're doing brain or something like that. It's best to pick a certain area of the brain, or you don't want to send in a whole brain because we can't process that. So if you have like a hippocampus or just anything else that you can isolate and that's what you want to look at, that's the best way to do it. For IP samples or pull downs, we don't really know how much is there, so that's really not. We just run everything that you give us, basically, and then, oops. For serum and plasma samples, like I said, we can go down to 10 microliters, but we like to have 25 to 50 microliters if possible. So when you're doing cultured cells, make sure to count the cells. Just don't kind of assume that you have that number of cells, because sometimes we'll get where you can't even see, this is what right down here should be about the size of 10 to the 6 cells. And sometimes you won't even see a pellet and they'll say that there's 10 to the 6 cells there. So
Assuming all cells.
yeah, that's true. I mean, that's just a normal size cell. Yeah. And as Dennis said earlier, make sure to wash the cells with PBS or something like that before, just because if we have fetal bovine serum or anything like that in there, it's going to be a contaminant. It's going to be the first thing that you see. It's going to be the most abundant thing. So it's kind of hard to normalize across. Sure.
So for cell culture cells, I usually leave my cells with trypsin already. I leave the cells with trypsin. So do you want the cells to be lifted with trypsin or scraped off?
You can use the trypsin to remove them from the plates. Just make sure to rinse them.
Okay.
Yeah.
Don't leave them in the,
Okay.
just don't leave them in the trypsin. Just make sure to rinse them with PBS. But that trypsin there is fine. Before you send the cells, make sure to snap freeze them immediately whenever you harvest them, and then store the pellets at -80 and avoid freeze thaw cycles. So just have them in the -80 until you're ready to ship them, and then you'll ship them on dry ice. And make sure to put them on plenty of dry ice, especially if you're not from Arkansas in the summer, and because it gets really hot here and the dry ice disappears very quickly. So make sure to put them on plenty of dry ice when you send them. And like I said, batch effects occur if you have cultures from different days. So that's where you would want to add that into the notes section of the sample list.
Karen,
Yep.
Can I send a lysate instead of cells?
Yeah. Yeah.
I
Yeah,
mean,
if you send the lysate, we're going to do a BCA on it just so we know before we start our sample prep. But yeah, you can send.
Let us know what the lysate composition is.
Yeah, like what the buffer or if it's in RIPA and
Yeah.
use protease inhibitors and things like that.
The biggest problem oftentimes is something like a reducing agent. Sometimes people, lyse their cells with DTT, and that's incompatible with current method we use, protein quant. So that becomes an issue. If we know it beforehand, we at least won't try it, right? So, but there are plenty of times when we've gotten a lysate in and we do BCA and they all turn black, and it's like, oh wow, this probably had some DTT in it. Sure enough, it was in there, but it wasn't in the list. It was only 10 millimolar. We didn't think it mattered. No.
actually, now I remembered, I should probably run a gel and send you.
No, that's always a great idea. I love, it's hard to argue against the visual confidence that running a gel gets you on protein amount. Yeah.
Plus the sizing, right, for the molecular weight. Yeah. Yep. Thanks.
Okay, so for tissue samples, fresh frozen ones, take 10 to, like I said, 10 to 100 milligrams of tissue. Perfuse or rinse with saline to remove any blood. Otherwise, you're going to get hemoglobin contamination in your proteomics, and especially if you, yeah, just make sure to rinse all of them. Sometimes we'll get one tissue that has, you can tell that there's still blood there, and then the rest of the samples don't have it. So the more consistent it is, the better. Again, size and uniform composition. We like to have that. Flash freeze immediately and store at -80 until shipped. This tube here is just kind of an example. Try to make sure there's no hair still attached to, because then if you have a lot of keratin contamination. So we do get samples like that every once in a while that still have hair on the tissue. So don't do that. Yeah, it's a huge struggle. So for plasma or serum samples, the amount of protein in serum is around 60 micrograms per microliter. So we, like I said, we can go down to 10 microliters, but we like to have more. Make sure there's no hemolysis, because that can also affect or be a contamination in your samples when we run them. Again, -80 prior to shipping. And then when we get those samples in, we run them through a kit that depletes the top 14 most abundant proteins from the samples before we actually go ahead and process them and run them.
You've put that to use commercially available collection tubes like Vacutainer. Do you have a preferred Vacutainer if it's human plasma?
I'm not sure. No, just any of the commercial. We just want to minimize as much lysis or hemolysis as possible.
So to avoid another freeze thaw, some people have sent us like their whole. We bought it, pulled out our last one, and then send it back. Because otherwise you're gonna unfreeze yours, make an aliquot, send it to us, we're gonna freeze it, then we're gonna unfreeze it again, so, you just have to decide.
Best to make your aliquots before you ever do any experiments, right? I mean,
Yeah, we gotta call it through. Okay, yeah.
That's another question. So if you have a sample left, you can ship back to us.
If you request it, you can.
Okay.
You can't do it if you request it 2 weeks later.
Yeah.
Yeah, you might want to request
No, it's already been picked up in the mail.
request that up front
Okay.
with Sam at the beginning. So the type of sample that we get will determine what type of protocol we use to collect the proteins and then do the digest. For sake of time, I'll just. So this is kind of an older picture because we no longer do tissue lysis this way, but normally we would take tissue that's in RIPA and protease inhibitor cocktail in the 1.5 ml tubes, and then we would sonicate it with a sonication probe to lyse the tissue. This is what we would do with cells, also with the tissue culture cells. And then we centrifuge it to get a clarified protein sample. Now we have a little better system for muscle and any other tissue. We have a bead beater that we can do 48 samples at a time, where we can just put them in with stainless steel beads and shake them really fast, and it breaks up the tissue and homogenizes it, and it makes it much quicker and more consistent lysis for us. Then once we have the lysis, or if you already send in lysates or anything like that, we'll go ahead and do a BCA assay so we can standardize the samples to each other once we have them. And then once we have that, there's a reduction in alkylation steps. So we want to break down the disulfide bonds with either using DTT or TSEP. And then we'll add an alkylating reagent such as iodocetamide to keep those bonds apart. So our go-to until just recently has been the chloroform methanol extraction, and we still do use this for different types of samples. Basically, there's the T-step that I was talking about. You would add that to the sample to 1 to 200 micrograms of protein. And then you would also add the iodocetamide at 60 minutes in the dark. And then we do a chloroform methanol PBS extraction, remove the aqueous phase, add in more methanol, and then our protein is this little wafer right here. It gets spun down to the bottom, and then we will take all the methanol that's left off of that, let it dry, add TEAB and trypsin, and we leave our trypsin on at 47 degrees overnight. We already talked a little bit about this, but for that we would have maybe 10 tissue or cell line samples. That's when we would do the CME, and then we would go in, do the trypsin digest, and then that's where they would get labeled with the TMT labels. And then they would be combined together, and then we would do the phospho enrichment if that's what we were doing for these samples. And then it would go through the fractionation and then be run on the mass spec. And Rick's going to talk more about that tomorrow. This is the workflow that we currently use for phospholipid enrichment. And this is one that we would do. We currently do it in the lab after we do the digest. So it would be one that you would pay us to do. So we basically have titanium oxide tips that we would run the peptides through. The phosphopeptides would bind to the titanium oxide, and then we would also take whatever came off of there through the washes. We would take off of there, and then we would run those also through a iron filter also to get out any phosphopeptides that had gone through the titanium tips. So then we combine these two together to get your full phosphopeptide enrichment. If you have pull downs or BioID or something like that that are on beads, we do a FASP or filter aided sample prep. And this is where we would do the digest on the beads, and then it would be on top of this filter, and then we would wash them, and then later we would do the trypsin digestion on these filters, and then we would rinse them through, and the peptides would come out the other side. So we've recently been working on higher throughput sample prep options. We have a Kingfisher Flex that can use magnetic beads to isolate the proteins. And this is a 96 well magnetic head here. These are just plates here, deep well plates that we can move the magnetic beads through these different buffers to collect the proteins. So that allows us to do 96 samples at a time. There's also a S-trap method. And I guess those are both S-trap there. So there's just ways that we can do 96 or higher at a time samples, which helps us go from doing 1,000 samples a year to 12,000 samples a year. So without adding too many more people. So once they've been run on the mass spec, we do quality control. This is just a sample of what Dennis will talk about later. It's a violin plot that's kind of a top view of your proteins for each sample. We want an equal load we try to get an equal load of protein across each sample, because the basic assumption is that the global proteomic abundance doesn't change that much, like the whole protein abundance. You're going to have smaller changes inside the global proteins. And so we want, like, this one doesn't look like the rest of them. We want them all to look pretty similar. So also, whenever we look at different proteins and see the differences throughout your different groups here, what we want is either we want big changes, then you can have a little bit more variability in your samples in the groups. But it's best if we if you have smaller changes between the proteins or between the groups, we like to have tighter sample variability. So this is a chromatogram of a normal lysate. Dennis has shown several of these before. This is one that is not quite so normal. And you'll see you don't have the broad high peaks. You've got these outliers here. And these are hemoglobin contamination. So it kind of, what you want to see is all of this stuff down here, but it's getting out competed by all the most abundant hemoglobin proteins. So basically just limit the variability in your lab before you send the samples if possible. Accurately measure the amount of protein in lysates or the number of cells or tissue that you send us, and communicate and ask questions at any point in time. Sam's always glad to ask questions or to answer questions, or if he needs to convey information to the sample prep people, he's really good about doing that too. And this is just a few samples of different samples that were not the best quality. And I think that's all I have. But now we are asked. Yeah. That was a half hour. I didn't rush this time. Go ahead.
I have a small question.
Oh, sure.
So, could you go back to the slide showing the quality of the protein, the valine?
Yep.
Yeah, this one. So you did this before you do the LCMS or after you got that?
After. So we have a meeting every week where we go over all of the projects that have been completed through the week. And then we look at all the quality control data just to make sure that everything looks like it was run appropriately on our end, to make sure before we send out data to the investigators.
Okay.
Just want to make sure everything looks good. So we look at all of the quality control data, and we look at all of the comparisons in your samples and things too, just to see if we can find anything that might be anything that we might have done that affected any of that.
So how do you like if something wrong with our samples, how do we know that it's not your fault, it's our samples problem before you go through that? You can only identify it after you run everything, or you can control it before you're loading the samples.
Right. So that's the whole reason we do this, is we know a lot of the things to look for if it's something that happened on our end. But yeah, we have to run the samples before we can tell how good they are. We can't tell if your samples, if it's something that happened in your lab until we run them.
Okay, got that. Thank you. And another question. So how many proteins could you identify between the 20 minutes and the 40 minutes run?
Can you hear that? How many proteins can you identify in the 20-minute versus the 40-minute run?
Yeah.
Yeah.
So it depends on the complexity.
Microphone? Sorry, just a second. He's gonna get a microphone.
It really depends on the complexity of the sample. And so if we look at a cell lysate, we might get 7,500 proteins in a 20-minute gradient, and in a 40-minute gradient may get 9,000. In some sample sets, that may be 8,000 proteins in a 20-minute gradient and 10,000 proteins in a 40-minute gradient. A serum sample is going to be, the difference is going to be much less. And so, you may get 800 proteins in a 20-minute gradient and 900 proteins in a 40-minute gradient. It's definitely, you reach a cost effectiveness somewhere, before you get out to an hour of separation. And it really depends on how interested you are in lower abundance proteins. And so you'll get the vast majority, in a really fast analysis, but, we shoot to try to get, 7,000 proteins in 20 minutes and 8,000 to 9,000 proteins in 40 minutes.
Okay. Okay, got that. So the tissues like the brain tissues, probably we can get like 8,000 proteins in 20 minutes
Brain.
and 10,000 in 40 minutes.
Brain tissue.
Brain tissue.
Yeah.
Yeah, so it's going to be variable depending on what region of the brain that you're looking at. But it's uncommon to get over 9,000 proteins in a brain sample
Okay.
without doing, more extensive, a significantly longer gradient.
Okay. Thank you so much.
Sure.
So I have actually a question about this violin plot as well. So when we have a situation like this, I guess we can normalize some of these outliers bioinformatically later on, but do you have any measure? I mean, can we kind of just treat differently the outliers to normalize them against the other ones?
No, go ahead.
You can. And so it's often the question of whether you can or whether you should. If the difference is merely an abundance issue, let's say that in the protein quant there was a compound in one of the samples that interfered with the ability of the protein quant to accurately quantify it, and let's say we thought we digested 100 micrograms, but we only digested 50 micrograms. In that case, bioinformatically, you can normalize, and the majority of those proteins you can essentially multiply by 2, in simplest terms, and get them up on the same level. But there are going to be some proteins down at the lowest level that you didn't detect. And so there's nothing you can multiply them by to bring them up, right? So for the majority of the proteins, you can do that. Most often what we see is not because of a protein quant abnormality, but there's something with the sample. So let's say it's a cell culture, and in one of these, the FBS from the cell culture media didn't get rinsed out as well. And so now we digested 100 micrograms of protein, but 30 micrograms of it was fetal bovine serum and 70 micrograms was your sample. If you analyze that, it's really hard to informatically normalize that back to the rest of the data set and have it make sense.
Okay.
So you can do it. It's likely going to be an outlier because it is an outlier.
So what do we do then in this situation? Is there like
in this case, I would throw it out.
Okay, so you can
the
throw it out.
argument for increasing the number of N's so that if there is a question, you throw it out, right? You can do a Grubbs test to see that you're not cherry picking the data going, well, if I take these 3 data points out, then my hypothesis is correct.
I see.
Yeah, you don't want to do that, but you can clearly see if something is an outlier, you can rationally throw it out, and increasing the N, increasing the number of samples is the key way to do this.
So is there any objective way to identify outliers that we need to remove? Like, for example, this looks really...
the statistical Grubbs test is my go-to, and if I had to go to the chalkboard and write it out, I would fail that one, right? But it's that's an easy test to apply that shows outliers.
But you guys would tell us, right?
Yeah, PCA visually shows that, and I don't know if we're going to show it.
No, I was also going to say that if we see something, that's when we do our meeting at the end of the week to look at all of them, that if that's something that we see, our bioinformatics people a lot of times will take that outlier out and do both with and without.
I see.
They'll do the comparisons with both.
Awesome.
And this is where if there's 7 or 8 in a group and you throw one out, you still get a lot of statistical power.
Yeah.
When you've got 4 in a group and you throw one out and now you've got 3, your significance doesn't change much because you've thrown out a clear outlier, but you've reduced your power by so much that oftentimes it doesn't help you as much as you might think it would.
And it kind of, you get another one that becomes an outlier
Yeah.
if you remove the one.
It's a great question.